Okay, let's go back to the slides. I have a feeling it's probably a test on that. Okay, so 1.3 levels of organization. All materials, non-living and living, are composed of chemicals. The human body is the sum of its parts, and these parts can be studied at a variety of levels of organization. Atoms are the smallest unit of a chemical. Atoms, okay. All materials are okay. They can be studied at a okay. You know you're starting on page 21, right? See the
Yeah,
slides
because
up
I was
there?
going through the slides and it would just kind of like
Oh, okay. Okay.
professor like introducing herself and
I just wanted to bring that up in case that was part of. Okay, so atoms are the smallest unit of a chemical. Um, all materials, living and non-living, are composed of chemicals. The human body is the sum of its parts, and these parts can be studied at a variety of levels of organization. Atoms are the smallest unit of a chemical. So if you look down in the picture, scroll down on your slide so you can see, she gave you a picture here. This is a lithium atom. This is one of the chemicals in the periodic table. It's got positive charges called protons. It's got um, neutral charges called neutrons. Those are the gray ones. And it's got tiny little electrons, these blue particles. It's got three. So usually the number um, of protons matches the number of electrons. So this is a different, sometimes they don't. You get different kinds of things when that happens. But um, this is just a picture of that. Okay, let's keep going. Atoms are the smallest unit of a chemical. Molecules consist of two or more atoms like those in this image, but they often have hundreds. So hydrogen molecules are two atoms of hydrogen that come together. That's a molecule. Macromolecules are larger particles composed of small molecules bound together. So if you have a bunch of small molecules and you bind them together, you get a macromolecule. Okay, that's a macromolecule. So here's some examples. Here's hydrogen molecules at the top. And then there's oxygen molecules down below. Okay. And what makes them different is the number of electrons that they carry here. Like these two oxygen molecules share bonds. Here we have water, which is a hydrogen molecule, another hydrogen molecule, and an oxygen molecule. I mean, an oxygen atom, sorry. Hydrogen atom, a hydrogen atom, and an oxygen atom. Together, we have a water molecule because there are three. There's two hydrogen atoms and one oxygen atom make a hydrogen oxide molecule. This is water. This is a water molecule. Okay, let's continue with that. Levels of organization. Macromolecules like DNA. Atoms are the smallest unit of a chemical. Molecules consist of two or more atoms like those in this image, but they often have hundreds. Macromolecules are larger particles composed of small molecules bound together, sometimes millions, like DNA. So what she's saying is DNA is an example of a macromolecule. Okay, let's keep going. Levels of organization. Cells. Cells are the basic unit of structure and function in all living things. So we have atoms, we have cells. Cells are the basic unit of structure and function in all living things. They're partly built of organelles. Organelles are smaller organs that are within cells. We have tissues and we have organs that are part of our larger body systems, but organelles are found inside cells, okay? Okay, let's continue. Levels of organization. Tissues like muscle or cartilage. Cells are the basic unit of structure and function. A cell is the smallest unit of life. Tissues are groups of cells that function together. So like skin cells come together and form tissue. Blood cells come together and form blood tissue. Okay. There's muscle tissue, there's skin tissue, there's different kinds of tissue. I'm sure she'll get into that. Okay, organs. Organs are groups of tissues with specialized functions, like your heart, your pancreas, your liver. Okay, these are groups of tissues that have special function. Organ systems are groups of organs that function together. So you have, I think there are 11, if I'm not mistaken, 11 organ systems. The cardiovascular system is the most important transportation system in the body. The nervous system is an important communication system. So the cardiovascular system, that's the heart and the lungs and the blood vessels, okay, the cardiovascular system. And it's outlined for you here, right? It's a transportation system. The nervous system is an important communication system. You will learn more about organ systems in lab 1. You will learn more organ systems in lab 1. Okay. All right. Organs are groups of tissues with specialized functions. We already saw that. Organ systems are groups of organs that function together. Organisms are composed of organ systems that function together, like this cat. And it has a system, it has lots of organ systems that function together to bring it together, okay? So now she's putting it all together. You have the atom, the molecule, the macromolecule that builds the cells. Cells come together and make tissues. Tissues make organs. Organs make organ systems. And a group of organisms, the group of organ systems make an organism like you, for example. You have different organ systems in your body. That's how that works. Homeostasis, negative feedback loops. Okay. So what she's doing here is she's giving you an example of a negative feedback loop. Okay. You've got the air conditioning heating system, right? And it's controlled by your thermostat. Your thermostat is that function um, that tells you when the air temperature in the house is too high or too low, and that triggers that to come on. So if it's too hot, let's say it's the summer, right? The temperature is too high, so the thermostat is high. That triggers the AC to turn on to lower the temperature. When the temperature lowers to a specific point that you set on your thermostat, then it will shut off. So now, in a similar way, what does your body do? Well, you have a thermostat, but not like an internal thermostat. You have an organ called the hypothalamus in your brain that measures when your body temperature is too cold. And so you shiver because your hypothalamus tells you, hey, I need some help. And you put on a jacket, you cover yourself with a blanket, whatever you do. But when your hypothalamus measures that your body temperature is normal again, you'll stop shivering. If what's making you shiver is cold, you can shiver for other reasons, but assuming that you're shivering because you're cold, you should stop when your body temperature is back to normal. Okay. So this is a diagram of that. You have a stimulus that comes from the bottom that goes to a receptor, and that receptor goes to a control center, and that's compared to your set point, compared to um compared to your body temperature or whatever system we're talking about. And then that control center sends a message out to the effectors, the muscles or the glands that control that, to take whatever action is necessary. Scroll up so we can see the rest of the. Okay, so then there's a change, there's a response that happens, and then it's corrected until another stimulus comes that stimulates the receptors, and then the receptors send a message to the control center. The control center says, hey, I'm too hot or too cold, whatever it is. And then those effectors take action to respond and change whatever needs to be changed. Okay. Why do we study chemistry in a class about the human body? Well, because there's chemical reactions that go on in your body. That's why. Elements are composed of atoms. Atoms of different elements vary in size, weight, and interaction with other elements. Okay, we've talked about that. With other atoms. And atoms are the smallest unit of an element. I think as many times as she's written that, probably going to be showing up quite a bit. Atoms are the smallest unit of an element. Okay, here's your picture of lithium again with its 3 neutrons, 3 protons. I mean, sorry, 3 protons and 3 electrons. Okay, so we're rehashing lithium here. Atoms are composed of subatomic particles. Now, here she tried to give you a little bit of a memory trick. So we have in the inside of the cell, which is this round circle, it's denoted by this round circle, we have positively charged units called protons. So proton starts with P, so does positively charged. Proton, positively, they're both P's. So proton is positive. An electron is negatively charged. Here's my electron out here on the outside. And a neutron, NEU, is neutral, meaning they don't have any charges. They're not positive or negative, they're just neutral. Okay. All atoms have protons in the nucleus, and almost all of them have neutrons there. The nucleus is the atom's central core. The electrons orbit the nucleus. So the electrons, this is a static picture, it's not moving, but the atoms fly around the nucleus, they circle around the nucleus. They're not static like this picture, but that's why they're on the outside. Okay. Since protons have a positive charge +1, and electrons have a negative charge -1, an electrically neutral atom has equal numbers of protons and electrons, which is what I told you, right? Then that the protons and the electrons have to balance each other. If atoms gain or lose one or more electrons, okay, they become charged and are called ions. Okay, so an ion is an atom that's charged, an atom that's not neutral. Could have a positive charge, could have a positive ion, that's called a cation, or you can have a negatively charged ion called an anion. We'll get to that. If an atom or a molecule has 11 protons, but 10 electrons. Okay, so here, this has 11 protons. So it has 11 positives and only 10 negatives. So it's got more positives, right? And it has a net charge of +1. And it's positively charged. This is just like mathematics. Positive 11 protons, and 10 negatives. So 11 positives, 10 negatives. They cancel each other, but you have one proton left. Protons are positive, so it has a net charge of +1. What if an atom or molecule has 9 protons, but 10 electrons? What do you think? What would that charge be?
Think it would be like -19.
It would be a -1 because this has 9 protons, so 9 of them are positive and 10 electrons. So this is like, let me see if I have, is it gonna let me rotate? One, I don't think it will. Maybe if no, it doesn't. Because this is, there's 9 positives and there's 10 negatives. So each of the positives cancels out one of the negatives. This is like +9 and -10. So the signs are different. So it would be a -1 for that. And that would be a -1. Okay, so let's continue here. Okay, so we're going to talk about atomic number versus mass number, which is chemistry stuff. So the element of an atom, the atomic number is the number of protons. So we've seen hydrogen before when we were looking at water, okay? The mass number is the protons plus the neutrons, okay? So atomic number is different than mass number, okay? Atomic number is just how many positive units you have, how many protons there are. The mass number is the protons plus the neutrons, okay? Together, because they're not always the same. Okay. Elements are substances that cannot be broken down into other substances. So if you look, you can do this with any element on the table. They highlighted carbon for you here. Carbon is the C, okay? This top number is the atomic number, okay? The number of protons. There are 6 protons in carbon, okay? The mass number is the number of protons plus the number of neutrons. So this is your proton number all the time. So since it's exactly double this top number, 6 times 2 is 12, we know that this carbon 12 has 6 neutrons because the mass number is the number of protons plus the number of neutrons. So 6 plus 6 is 12. There are 6 neutrons in here. Okay. Know the symbols for the 12 elements critical to human life. Oxygen. Oxygen is O. Okay. It's next to carbon. It's a couple of carbon they gave you. Carbon is over here. Okay. Hydrogen is on this first column at the top. Nitrogen is next to carbon. So carbon, nitrogen, oxygen are all right together. Okay. Phosphorus is below that in the next row, below nitrogen. That's phosphorus. Sulfur is next to that. S. Calcium is over here in the second column on the left. That's Ca, like California. Chlorine is Cl. Chlorine is down here on the other side of the table. It's under fluorine. Okay. So you've got carbon, nitrogen, oxygen, phosphorus, sulfur, chlorine. Magnesium is Mg. It's in the second column over here on the left. Don't confuse magnesium with manganese, Mn over here. Potassium, and then sodium, and then iron. Those are tricky because potassium is not P, okay? Potassium, I believe, is K, if I'm not mistaken. Sodium is Na, and iron is Fe, because they come from other languages. So there's iron is Fe, and we're going to talk about that. But those are your elements: oxygen, carbon, hydrogen, nitrogen, phosphorus, sulfur, calcium, chlorine, magnesium, potassium, sodium, and iron. Okay? Go ahead and scroll up. I heard oxygen and magnesium are dating. That's going to be explosive. I was like, oh my God. When you combine oxygen and magnesium, you get an explosion when you do that. Okay, let's continue. Electrons do not orbit the nucleus randomly. They are confined to specific orbitals called electron shells, okay? So I don't know if you remember, but when we were looking at water molecules have 8 electrons, and they need 2 shells to hold that, because your first electron shell can only hold 2 electrons. That's all you can hold in your first shell. So if you need more than 2, you have to get a second shell, and you might need more than that. The outermost shell can hold 8 electrons, and this gives you some configurations. Here's a cell, an atom with 1 electron. Here's a cell with more than that, and you've got more electrons on the second shell on the outside. But these are just different configurations of the same, okay? So here's hydrogen. Here's carbon with its 6 electrons. 2 on the first shell, and then you need another 4. So it has 1 at each direction. That's carbon. And then these are empty. These out here are empty. Nitrogen has 7 protons and 7 electrons. So again, you need 2 on the first shell here. But then the second outer rim, you need another 5. So 2 at the top, and then 1 at each of the other cardinal directions. Oxygen is easier because it has 2 in the initial shell, and then it has another 6. So it's got 2 at the top, 2 at the side, and then it shares 1 and 1. Is usually how it's drawn. Okay, that's an oxygen atom. Now, magnesium has an atomic number of 12. So there's 12 protons, 12 neutrons, because they have to match. And so you only have two. Now, the reason that she's saying how many are in the outer shell is, remember, she said that the outer shell can only hold 8. But if the first shell can only hold 2, like this does, then how many are in the outer shell? Well, remember, the second shell can hold 8 more. So that's the 2 that are on the inside, and then the 8 that are there, right? So what would happen to the ones that are missing? Let's see what she said. Does she say? Let's go there. For practice, try these, okay. Okay, she's having you draw electron diagrams for helium, neon, sodium, aluminum. Okay. Did she have you do these as exercises, or?
No, not really. She just like went over like this part and then
She went over which part? Oh, she
um
went over what the electron shells are like.
yeah, but she didn't like ask us to
Have
do
you
like
do
that
that?
in class.
Okay. Um, well, maybe we should, hold on, let me see. Electron. Let me see if I can. Because I think that would be useful to at least, I'm gonna go ahead and pause. Because she's probably gonna have you, um, she's probably gonna have you draw a, if not draw it, identify. You can go ahead and bring your slides back at any time. And then but you could go through and figure out how many are gonna be in each. She'll probably have you memorize a few levels, how many are in each one, so you'll know. But go ahead. What else are we doing? Okay, so what is the atomic number of this element? Okay, well, the atomic number is the number of protons that it has. So this would be 13. But its mass number is 13 plus 12. 13 plus 12 is 25. So we have 25. Okay, let's keep going. I'm sure she shows us something about it. What is the atomic mass number of this element? Atomic mass number here is 13. Again, it has 13 protons. Okay, so notice that the last two that we've looked at have had 13 protons. What's different is the number of neutrons. This has 14 neutrons, so its mass number is 13 plus 14, which is 27. So this is what we call an isotope for this element. It has different numbers of protons, I'm sorry, different numbers of neutrons, but the same number of protons. Okay, keep going. Isotopes are atoms with the same atomic number, but different mass numbers. Okay? Okay, so that's what we just saw there. What is a hydrogen ion? Okay, well, hydrogen has an atomic number of 1. We know that. So if the atomic number is 1, how many protons are in the nucleus? Tell me, how many protons are in the nucleus?
I think it would be like 5.
Why do you think that? Tell me why.
Mhm.
The atomic number is the number of protons you have. So if I tell you the atomic number is 1, there's 1 proton in the nucleus, okay? It doesn't matter how many other things you might see floating around. The atomic number matches the number of protons, okay? So there's hydrogen has an atomic number of 1. There's 1 proton in the nucleus. How many electrons will it have if the atom has no charge? So if the atom is neutral, how many electrons do you need to make it have a 0 charge?
I think it would be 0.
Okay, that's a 0 charge, but let me show you something. Let's try this. Neutral means that it's not charged. That's true, but that's not the question that I'm asking you. What I'm asking you is this. Let's try this. So let me go here. And let me share this. Now, I'm going to replace that and I'm going to share this. Now, I can annotate the screen. Okay, so here is a, well, it would be if I could figure out what I'm doing here. There it is. All right, so here's my, okay. Now, inside, I have another circle in here. Like that. Okay, let's say here. Should be in the middle, but let's humor me. So if I've got 1 proton in here, okay, so that means that there's a positive 1 that comes here. So this is going to be let's see. This is going to be a plus 1 in here, okay? Now, that's a plus 1, okay? If I want to make this balance, then I need an electron out here on the outside. Because remember, this shell can have 2. So I need an electron that has 2. So here, or that can take up to 2 if I need it. I won't need that many. But here, I've got let me make another circle here. And let me make it in yellow so that matches. So if I do this, here's my electron over here, okay? Now, if it's balanced, then yeah, I have to make this charge 0. So in order to make that charge 0, this would have to be a -1, because here, this is a -1 out here, okay? So -1. +1 and -1, if I write, if you think of it like this, and I say +1, oops, +1, and then I go +1, and then I say I have a -1 out here. This is a -1. +1, -1, that's going to give us 0, like you said. What I was looking for you to tell me was what is, yes, I want this to be 0. But what I was looking for you to tell me is if I tell you this is +1, I need 1 electron out here. If I know this is neutral, if I know this is 0, what electron do I need here? Which is I need a -1. Now, if I run up on another molecule, let's say, and it gives me an electron, then I become charged, because then I have more electrons out here than I do in here, and I can't balance anymore, right? So that becomes an ion. But right now, this is a stable atom because it doesn't have a charge, right? And it's a hydrogen, okay? So that's a stable atom because the charges are balanced. So you would need 1 electron to balance this. I'm gonna take a screenshot of that, okay? All right, let's stop sharing so that I can go ahead and put up the screen share again, and then I will. Okay, so hydrogen has an atomic number of 1. How many protons are in its nucleus? 1. How many electrons will it have? If the atom has no charge, it will have 1. What does a hydrogen atom look like when it loses its electron? Well, then it becomes positively charged, and it becomes a cation. So let's continue. Maybe she hasn't told you what the name for that is, but we're getting there. Okay, now we're going to talk about acids, bases, and the pH scale. Acids are chemical compounds that donate hydrogen ions. Notice this is an H+. This means that your hydrogen ion is charged now. it lost its electron, okay? This is what happens to it. So this hydrogen, which is H, is now an ion. It's positively charged. So it lost its electron because it donated it and it became an acid. So a hydrogen atom that loses its electron becomes an acid. A base is a compound that accepts hydrogen ions, removes them from solution, and leaves behind excess. This is OH. You can read that as OH minus. But this is, the word for this is hydroxide when you read it. So leaves behind excess hydroxide ions. The pH scale describes the acidity of a solution. So how many hydrogen ions did you donate? If you're very acidic, like hydrochloric acid, you're like a 1. Cow's milk is almost neutral. Human blood is almost neutral. Things that are basic are things like egg whites are slightly basic. Milk of magnesia, if you've ever taken that medicine for your stomach. Anything that if you have an acid stomach, if you've heard that, when you have too much acid in your stomach, they'll have you take something like milk of magnesia or Pepto-Bismol. Those are basic compounds, not basic like simple, but basic like removing acid from your system. So the scale goes from 0 to 14, okay? Your acidity level will increase as the number gets lower, okay? So you have an acidity, an acid level. When your pH is low, you have a base level when you're neutral at 7. So anything below 7 is acidic. Anything above 7 is basic. Okay. Okay. Buffers are substances that resist changes in pH. So they're the ones that try to stay neutral, try to stay as close to neutral as we can. They accept hydrogen ions when they're in excess, and they donate hydrogen ions when they're depleted. Okay. And these are just some examples. This is another example of the table. Important bonds in biological molecules: hydrogen bonds, ionic bonds, and covalent bonds. Now, these are the 3 ways that you can bond. There are more, but these are the 3 basic ways that molecules come together and attach themselves to other molecules: hydrogen bonds, ionic bonds, and covalent bonds. So hydrogen bonds are bonds with water molecules or hydrogen molecules. This is water. Water is a very famous hydrogen bond because hello, water is 2 hydrogen and 1 oxygen. So that's your hydrogen bond. Ionic bonds form when you have molecules that are charged, usually at least 1 positive ion and 1 negative ion, sometimes more than 1 positive and more than 1 negative, but this is a common ionic bond is table salt. You've got sodium chloride. This is a positively charged sodium with a negatively charged chlorine molecule. You mix them together or you bond them together, you get table salt, okay? That's an ionic bond. Then you have covalent bonds. Covalent bonds they can happen between 2 of the same molecule or different molecules. But here, they don't just combine their stuff, they share. They share molecules. So when they share something, when they share 1 or more electrons, you get a covalent bond, okay? Because they're sharing those, what they call valence electrons. We'll get to that a little bit later. But the valence are those shells that we drew earlier. They're sharing valence electrons with each other. Okay? Covalent bonds are formed when atoms share electrons in order to fill their outermost shells and become stable. A covalent bond is the sharing of a pair of electrons by 2 atoms. Okay, so they share a pair of electrons, not just 1 electron. They have to share a pair of electrons by 2 atoms. Okay. So here we have, now here's an example of covalent bonds between 2 hydrogen atoms. Here's hydrogen, here's another hydrogen. Notice they each have 1 electron. They come together, right? And they share this pair of electrons in this outer shell. This becomes H2. This is a hydrogen molecule. Still hydrogen, but it's not H1, it's H2 now because it's a hydrogen molecule. It's 2 hydrogens together, okay? Each water molecule is put together by 2 polar covalent bonds. Oxygen is a bully. So here you've got hydrogen with its 1 electron, right? Hydrogen has its 1 electron. The oxygen has this negatively charged, partially negative, right? So the hydrogen molecule is sharing between its electron and the oxygen, 1 of the electrons from oxygen here. The electrons are pulled toward oxygen, okay? So basically this oxygen comes along and picks up these hydrogen. That's why this oxygen looks so much larger, but they attach at the poles. They attach at the bottom of the oxygen molecule, okay? Okay. Covalent bonds may be polar or nonpolar. In nonpolar covalent bonds, the pair of electrons is shared evenly. Each atom in the bond gets the pair of electrons for half time. Kind of like the two hydrogens, they share nicely, right? In a polar covalent bond, one of the atoms is cheating and keeps the pair of electrons for more than half the time. The atom in a polar covalent bond that keeps the electrons more than half the time has extra negative charge. Now, if you have a polar covalent bond, right, that polar covalent bond is polar because it's pulling on that other molecule or that other atom. It's pulling electrons. So if it's pulling electrons, it's pulling negative charge, right? So it's going to have a charge on it, and that's why it's negative, because that atom is going to be balanced, is pulling negative charge toward itself. So it's going to has extra negatives. The atom in a polar covalent bond that has the pair of electrons less than half the time ends up being slightly positively charged. Polar covalent bonds are necessary, are necessary for the formation of hydrogen bonds. Yeah, okay. Okay, so now we've talked about hydrogen bonds, covalent bonds. What about ionic bond? Well, in ionic bonds, you have one atom that loses electron because they donate their electron, or they give up their electron. When you give up an electron and you're stable, you end up having less negative charge than you did. So you become positively charged. Another gains the electron and it's going to become negatively charged. And then the difference between the charges makes them come together, makes them bond together. So here she gives you a picture of that. The outer shell here on sodium has 1 electron. The outer shell here has 7 electrons. Remember, it wants to have 8. So, and it would like to have an outer shell that's complete. This outer shell is missing an electron. This outer shell only has 1 electron. If I got rid of it, it could have 2 shells that were full. So the sodium is going to give up its electron. That means it's going to become positively charged, and it's only going to need 2 shells instead of 3. You see, we have 2 shells here instead of 3. And the outer chlorine shell is going to now be stable because it's going to have its 8 electrons, so it'll be slightly negative, and we get a very stable sodium molecule as a result of it. Okay. Charged atoms are typically called ions. Ionic bonds form between oppositely charged ions. Hydrogen bonds are found between water molecules. An atom in one polar covalent molecule is electrically charged or electrically attracted to the oppositely charged atom in another polar molecule or in another part of the same molecule. Okay. So if you have a polar covalent molecule they're attracted to the oppositely charged atom in another polar molecule. Okay. Hydrogen bonds are the weakest type, but there can be lots of them, and that's what she's showing you here. This is a very small part of a molecule of DNA. Each molecule is depicted as a colored sphere. So you can see how many different colored beads there are in just this small sphere. One of your DNA molecules is built from hundreds of millions of atoms. It is actually two long chains wrapped around each other to make a spiral staircase form known as the double helix. The two long chains or strands are joined to each other only by hydrogen bonds. Okay. Now we're going to get into writing formulas. A molecular formula represents the number of and types of atoms. For example, glucose equals carbon 6. This means there are 6 carbons because carbon is C. There's 12 hydrogens and 6 oxygen atoms. So glucose is C6H12O6. Water is H2O, 2 hydrogen atoms and 1 oxygen atom to make 1 molecule of water. Okay. Various representations called structural formulas can be used to illustrate how atoms are joined and arranged in molecules. Structural and molecular formulas. So a structural formula uses single lines to illustrate single bonds, double lines to represent double bonds, etc. So you can use a structural formula like this, or you can use three-dimensional molecular models. Go ahead. And these are typically manipulative. So you have a water molecule with a hydrogen, and an oxygen attached. And again, here we have the oxygen being the bully, right? So it's sharing unequally with this hydrogen. Okay. A chemical reaction occurs as bonds are formed or broken between atoms or ions or molecules. Okay. Substances that are changed by the reaction are called the reactants or the substrates, the left side of the arrow. The reactants or the substrates. The substances that are formed by the reaction are the products. So the reactants turn into products, or the substrates turn into products. During synthesis reactions, 2 or more atoms or molecules bond together, forming a more complex product, like table salt, for example. A plus B equals AB. During digestion reactions, larger molecules are broken into smaller ones by breaking chemical bonds. AB breaks down into A plus B. Carbon is an exceptional element. It is the only molecular basis for life. All the other molecules that make life possible are based on carbon. Okay. Organic molecules make life possible. So organic molecules are molecules that have carbon or they're derived from carbon, okay? Hydrogen and other elements are covalently bonded to carbon. Carbohydrates, lipids, proteins, nucleic acid. Okay. Polymers and monomers. Polymers and molecules are made of smaller repeating molecular subunits known as monomers. Think of the polymer as being like a long train. It's made up of a variable number of train cars. The train cars would be the monomers in this analogy. Right? I'm sure she's going to give us more examples of that. Carbohydrates are the simplest biomolecules. Carbohydrates are the simplest. So now we're getting into, she's covered a lot of ground because we're going into organic chemistry here and some biochem. Carbon and hydrate water. The building blocks of carbohydrates, sugars. So basically you have monosaccharides, you have disaccharides, you have trisaccharides, you have polysaccharides. That's just how many chains you have going. So monosaccharides are monomer. They're single chains of glucose molecule. Okay. They're the single chains of carbon. So some glucose molecules, like this one, have a straight chain of carbon atoms. There it is right in the middle. Here's your hydrogen and oxygen with your carbons attached. 1, 2, 3, 4, 5, 6. 6 carbons just attached to it. More commonly, glucose molecules form a ring structure. So here's more commonly what they look like. You have carbons on each vertex of the shape, for example, except for one. And then you have hydroxide. This shape symbolizes the ring structure of a glucose molecule. Okay? Okay, what I was just telling you, mono, di, and polysaccharides. Sugars are fast energy. Complex carbs are slow energy or structure. The monomers of carbohydrates are sugars or monosaccharides. Notice, monosaccharide, you have 1 glucose molecule. Disaccharide, you have 2 glucose molecule. Disaccharide, this is saccharide here. Mono means 1, di means 2, and there's 2. And then if you scroll up, I'm pretty sure this is going to be poly. Okay, so this one has a whole bunch of glucose molecules. Got 3, 4, 5, 6, 7, 8, 9. 10, 11, 12, 13, 14, 15. This has 3, this has 6, and this has 6. So this is a polysaccharide example. Okay. Not all carbohydrates are digestible by humans. Ingestible carbohydrates in food is referred to as fiber. Okay? This is this is our friend the spiny lobster here. So far, carbohydrates, sugars are the building blocks, monomers. Simple carbs are monosaccharides and disaccharides. Complex carbs are starches, glycogen, cellulose. Okay, cellulose we can't really digest. Cellulose, like in this, like if you've ever eaten celery and you have that green stringy part that you really can't chew on, that you have to kind of throw away, that's cellulose. That's not very digestible for us. But that's okay. Fats, lipids are also organic molecules. Fats are made of carbon, hydrogen, and oxygen, just like carbohydrates, but have fewer oxygens and more hydrogens for each carbon. Fats do not dissolve in water because they are nonpolar. Fats, lipids, are not really polymers, but they're made of fatty acids. They're often made of fatty acids. Okay. Fats, okay, we said that. So now they have a carboxyl group on one end, a carbon backbone, and you can have fats that are saturated or unsaturated. Structure of triglycerides. Each molecule of triglyceride is made up of 3 fatty acid molecules attached to 1 glycerol molecule. Whoops. Hold on. Triglycerides are the most common fat. Triglycerides are the best way for animals to store energy for the long term, because we can put more calories of energy into the lightest form. Okay. Other types of lipids are phospholipids that consist of glycerol, 2 fatty acids, and a phosphate group. They're important in cell structures such as plasma membranes. Phospholipids are similar to triglycerides. Cell membranes are made up of phospholipids. They only have 2 legs or 2 projections, and they are amphipathic. I don't know what that word means, but I'll find out. Let me So certain molecules, these phospholipids, they have two branches, they have two legs. They have a hydrophilic head. They have a head that loves to be in water. And they have hydrophobic tails, tails that do not like to be in water. They don't. So they're amphipathic, meaning they have two loves. Their heads love water and their tails hate water. Okay. So they're amphipathic. So the extracellular fluid is the watery fluid outside of the cell, and the intracellular fluid is the watery fluid inside the cell. Hydrophilic heads extend toward the intracellular and extracellular fluid, and hydrophobic tails are directed away from that. Each molecule of phospholipid is like one kid in an inner tube at the world's most crowded water park. So you get too many phospholipids and you don't really get anywhere because it's too crowded. Now, we're going to talk more about the cell. The plasma membrane or the cell membrane separates the living cell from its non-living surroundings. It is made beginning with a phospholipid layer. It's got hydrophilic head and a hydrophobic tail. So outside the cell is hydrophilic because you want that hydrophilic head to be facing where the liquid is on the outside of the cell. They love water, they love liquid, so they're going to have their heads toward the outside of the cell, and they're going to have their tails on the inside, which is what you see here. Now, other types of lipids, you have phospholipids that consist of glycerol, two fatty acids, and a phosphate group, like they're important in things like the plasma membrane. The other types of lipids are things like steroids. They have complex carbon ring structures. A very important steroid is cholesterol, which is used to synthesize steroid hormones, including testosterone, estrogen, progesterone, cortisol, and aldosterone. Steroids. Okay, she hasn't told you much about steroids. I'm sure we'll get into that depending on where she's going with this, but that's where we are. Carbohydrates are sugars that are made up of sugars that are the building blocks, the monomers. Simple carbs are monosaccharides and disaccharides. Complex carbs are starches. Then you have fats, which are lipids. Fatty acids are the building blocks. Triglycerides are a storage form of energy. They're hydrophobic. And then scroll up. Phospholipids form the cell membrane, and they're amphipathic. They have those hydrophilic heads and hydrophobic tails. Amphipathic. Okay. Now we have proteins. So what she's doing here is she's going through the four basic types. She's going through carbohydrates, lipids, proteins, carbohydrates, fats, proteins, and basically is what she's doing. Proteins are organic compounds containing carbon, this is carbon, oxygen, hydrogen, and nitrogen atoms. Some also contain sulfur. Okay. Carbon, oxygen, hydrogen, nitrogen. Proteins have a great variety of functions in the body as structural materials, energy sources, certain hormones, receptors on cell membranes, antibodies, and enzymes to catalyze metabolic reactions. Amino acids are the building blocks of proteins. Proteins consist of building blocks called amino acid, each of which contains a carboxyl group, an amino group, and a side chain called the R group. Most living organisms have 20 different amino acids making up the protein. Amino acids bind together in polypeptide chains of less than 100 to 5,000 amino acids. So again, it depends on how many amino acids you need. Proteins are much more complicated than carbohydrates or fats. To make a more complicated structure, we need to add nitrogen. You have carbon, hydrogen, and oxygen. If you want to make it more complicated, then you add nitrogen to that. Proteins are polymers built from monomers called amino acids. There are 20 different amino acids in most of life. Each amino acid has a central carbon atom bonded to a carboxyl group on one side So this is just showing you the different, and an amino acid on the other side, and a side group that is variable among all 20. So the amino acids that build proteins differ most by their carboxyl group and their R group. And these are just some of the different amino acids that you can see there. Peptide bonds link one amino acid to the next. Amino group, carboxyl group, amino group, carboxyl group, amino group, carboxyl group, connected with this carbon hydrogen side group. The name of the covalent bond that connects, keep going. The name of the covalent bond that connects one amino acid to the next in a protein is a peptide bond. This is why small proteins are called peptides. Large proteins are often called polypeptides. So it's a covalent bond. So remember, they're sharing a pair, at least a pair of electrons, right? And that sharing, if you have a small chain, you get a peptide. If you have large chain, they're polypeptides. There are 20 amino acids in humans. 5 are charged. 4 are polar but uncharged. So there's 5 that are charged, they're ionic. 4 are polar, so they have to do with water, but are uncharged. 8 are non-polar, so they don't deal with water. And 3 are special amino acids. The side chains interact with each other. If a positively charged side chain is next to a negatively charged side chain, they will snap together by an ionic bond. If two positively charged ones are next to each other, they push away from each other. So now we're talking about the levels of structures in proteins. Primary structure is the sequence of amino acids. The secondary is the domain they have, or the type of shape they have. They're pleated sheets or alpha. And the tertiary is the overall 3D structure of a protein. What kind of machine or structure is it? And I'm sure she'll get into that. Okay. Quaternary is not all proteins. Muscle proteins that are multiple proteins that are joined. One muscle protein is made up of thousands of individual protein subunits. Protein conformation and denaturation. The conformation is a unique three-dimensional shape of a protein due to its hydrogen and covalent bonding in different parts of the polypeptide chain, which determines the function of a protein. So depending on where the bonds are in the protein and what type of bonds you have, you get different kinds of protein. And that's how you distinguish the shapes, where they bond and what kind they are. Denaturation is irreversible disruption of a protein shape. So one way to denature protein is with radiation. So people who have chemotherapy, for example, that have a lot of radiation suffer from a lot of denatured proteins. Now, some of those proteins are feeding the cancer cells. Some of those are healthy proteins, and some of that denaturation can't be reversed. Those cells die. When another form of denaturation is radiation poisoning. When you have X-rays or you have like a that they can't do their jobs. Okay, they're caused by pH changes, excessive temperature changes, radiation, or chemicals. Here's an example. If you think about cracking an egg, you have the white part, you have the yellow part. You cook the egg long enough, the albumin becomes denatured, so it changes its form. Cooking an egg denatures the albumin, the egg protein. Doesn't make it bad for you. That's just an example. It's bad for the egg, but it's not bad for you. She's just showing you that that's an example of how heat denatures protein. Okay, let's continue. Some proteins are structural, but most are amazing machines. And here's some examples of pictures of proteins. Enzymes are just one example of protein machines. So here's ATP and ADP. This is your energy molecule in your body. They are made in the mitochondria of your cells, which you may or may not remember from biology class. But this is just more examples of protein machines. Okay. So we're doing a recap again. Carbohydrates, we have sugars are the building blocks, the monomers of carbohydrates. Simple carbohydrates include monosaccharides with one chain and disaccharides with two chains. Complex carbohydrates are starches. Fats or lipids are made from fatty acids. Triglycerides are a strong form of a storage form of energy. They're hydrophobic. Phospholipids form the cell membrane and they're amphipathic. So they have the hydrophilic head and the hydrophobic tails. Okay. Proteins are made from amino acids. Proteins are machines that can also be structural. That's what we just kind of recapped with that. Organic substances are things like nucleic acids. They form genes and they store information for protein synthesis because genes are protein, basically. They can be enormous organic molecules. They contain carbon, hydrogen, oxygen, nitrogen, and phosphorus, okay? Nucleic acids are polymers made of monomers known as nucleotides. Each nucleotide consists of a 5-carbon sugar, a phosphate group, and one of 5 nitrogenous bases. So sugar, a phosphate group, and one of 5 bases. We're going to talk about that, I'm assuming. Nucleic acid molecules are chains of nucleotides. DNA is an enormous double helix, 2 strands twisted, which she's told us already. And so here's a picture of your 2-stranded double helix. RNA is usually single-stranded and has a structure like a protein. That's usually one of the reasons, one of the ways, it's not the only way, but it's one of the ways that RNA typically is different from DNA. DNA is 2-stranded, and it's a double helix. RNA is usually single-stranded, and it has different functions. Okay, so let's continue. So here's some some pictures of nucleic acids. Nucleic acids are polymers of nucleotides. So you've got, here's phosphate in the middle of this, or phosphorus, with a bunch of oxygens attached. So 3 of the oxygens are attached by single bonds, single oxygen bonds. This phosphate and this oxygen have a double bond. That's what the 2 lines mean. So now you've got this base, and you're gonna get into that. The bases adenine, guanine, cytosine, or thymine. In RNA, one of those bases gets replaced with uracil, if you're wondering where the 5th base comes in, but DNA has 4 of these, and sometimes one of them gets replaced for the 5th one. So that's why that's 5. But this is your basic molecule. You've got double carbon bonds. Here you've got carbon-oxygen bonds. Here you've got a lot of helium bonds outside. Okay, these are different nucleotides. This is thymine. You can scroll down and you can see you've got there's a basic sugar. This is deoxyribose. It's just basically carbon and hydrogen. So we're back to the same slide. Sugars are the building blocks of carbohydrates. Simple carbohydrates are monosaccharides and disaccharides. Complex carbohydrates are starches. Fats or lipids. Fatty acids are the building blocks of fats or lipids. Triglycerides are a storage form of energy. They're hydrophobic. They hate water. They hate liquid. Phospholipids form the cell membrane, but they have a hydrophilic head, hydrophobic tails. They're amphipathic. And the amino acids are the building blocks or the monomers of protein. For nucleic acid, we have nucleotides as the building blocks, and nucleic acids form genetic material, like DNA. So, here's a couple of questions for us. An atom with a negative charge has more protons than electrons, more electrons than protons, more neutrons than protons, or more electrons than neutrons. If it has a negative charge, it has to have more negatives than positives. So which one would that be?
I think it would be more electrons than protons.
That's correct. More electrons than protons, because the neutrons aren't going to do anything. They're neutral. That's right. Is there another question? Which of the following is an example of a polysaccharide? A polysaccharide, that I don't know. Polysaccharides have their bigger sugar. Glucose is a simple sugar. So glucose, starch, maltose, or fructose. hold on, let me. Which of the following is an example of a polysaccharide? Those are starches, because glucose is a simple sugar. Fructose is made up of two. And I believe maltose as well is made up of two sugars. Okay. More question? What are what are saturated fats saturated with? In other words, what makes them saturated? Phosphorus, nitrogen, carbon, or hydrogen? So she's asking you what makes saturated fats saturated. So that is going to be, so they're large proportions of fatty acid molecules. And so that's going to be depending on how many they have there. So let me go back. Okay. They're hydrogen molecules. Saturated fats are saturated by hydrogen molecules. Saturated fats are saturated with hydrogen molecules. And let me read this into the recording. Let me actually, you know what? Let me do this. Let me put it here. And you can, I'll put it in chat too. So here it says, there's your. So what makes them saturated is the hydrogen, because they're separated by carbon molecules. If they're not, saturated fats are saturated with hydrogen molecules and contain only single bonds between carbon molecules. So that's that part. What's next? What are the major lipids of plasma membranes? The major lipids of plasma membranes. In other words, what are they made from? Triglycerides, phospholipids, fatty acids, or steroids? Major lipids of plasma membranes. So here, what are the major lipids of plasma membranes? They're phospholipids, okay? Phospholipids are the major lipids found on the plasma membranes of cells, okay? So that's where we are with that. And that's, I guess those are some answers for the other part, okay? And then we have another one. Isotopes of atoms, okay. Isotopes of atoms differ in their neutrons, electrons, protons, or atomic nuclei. Okay. Well, isotopes have the same number of protons, but they have different numbers of the atomic nuclei just means the atomic number. So what makes them different is they have a different atomic number because they have a different number of electrons. Isotopes of an element due to the number of neutrons. Okay. They have different numbers of neutrons in the nucleus and a different number of, that's what makes and that was in your slide on so isotopes have the same number of protons, but different numbers of neutrons. For the same element. So the answer there would be neutrons. Okay, they differ by neutrons. I typed it in the chat for you. Okay. Okay, Daniela. I think we're done for today, right?
Yes.
That was a lot of slides. Just to let you know, I did get you your 11 o'clock on Monday, but I have to be done by 12:30. Okay. Because
Okay, that's fine.
I have an appointment at 1 o'clock. That was a lot of slides to go through. That's probably why it's not due. Your quiz is not due till the 11th of February. Did she finish the whole deck this week?
No, she really didn't. She just went kind of like through half of it,
I
and
was
then
gonna say
like
because that's a lot of that's
yeah.
probably like that's a big chapter to cover all that. That's all your biochemistry in one chapter, pretty much. How do you like the teacher so far?
Well, I think she's okay.